may fall asleep before you do. So is talk about some of the work we've been doing specifically on, let me see if I can get a pointer here, on using nanobiotechnology to increase the stress resilience of different crop species. Some of what we were just hearing about reactive oxygen species is going to be very relevant to what we're talking about here. Now, I noticed in looking at the agenda that I'm actually one of a very small number of people talking about nanotechnology. So I think in terms of the way to think about nanotechnology is basically when you have a material that's at the nano scale, in other words, the particle size is below 100 nanometers, that material, most often we're going to be talking about nutrients, that nanoscale nutrient behaves differently, both physically and chemically, and also even biologically. So that's a behavior that we're going to take advantage of. So the nanotechnology program at the Connecticut Agricultural Experiment Station in the US is about 15 years old. And the project actually started in nanotoxicology. My PhD is in environmental toxicology. So when I started working with engineered nanomaterials, it was mostly out of a concern that these materials were going to have a negative effect on crops, on agriculture. So we started investigating a lot of different questions related to how these materials were going to negatively impact plants. And what we noticed as we got into this was that reactive oxygen species, ROS, were a critical part of the toxicity of these materials to plants. But what we also noticed after a number of years was that under certain circumstances, these materials were actually not only not toxic, but they were conveying benefit to plants. So that's when this applications-driven research started. This work's funded mostly by the US Department of Agriculture, although I'm part of two centers, which I'll mention briefly. But the other interesting thing that we noticed is that in some of the instances where these materials were conveying benefit to plants, reactive oxygen species were also really important. So we started thinking more broadly about reactive oxygen species. And we published this paper in Nature Food in 2022. And a lot of this was looking retrospectively at a lot of the data that we had, specifically around ROS, reactive oxygen species. And as we just heard, these are metabolites that are generated. They're generated actually during basic cell metabolism, but they're also generated under stress, including exposure to xenobiotics. And as we know, these reactive oxygen species, if the concentration's too high, they're going to damage biomolecules, organelles, so forth and so on. So cells, plants, animals, bacteria, otherwise, we've evolved pathways to kind of quench these reactive oxygen species, and those are some of the pathways you were just hearing about. But what's also important is that these reactive oxygen species are also signals. It's a signal to the plant that a stress is happening, and then the response to that signal is a significant upregulation of all of these stress-related pathways. So we started thinking about this in two different ways. And that's what you see up here on the right. So can we use nanomaterials that are able to scavenge reactive oxygen species? And can we also use nanomaterials that are actually going to trigger the reactive oxygen species and the subsequent response? Can we use that together to basically control homeostasis in these cells, in these organisms? And can we use that for our benefit? Because we know that if we're upregulating these stress response pathways, that's going to convey tolerance to abiotic and biotic stress. So this first pathway, which we have a lot more data on, is recovery. That's ROS scavenging. Plants exposed to something, it causes stress. We can supply a nanoscale micronutrient to alleviate that stress, to scavenge those ROS. What's interesting, and I'll finish with this data in about 15 minutes, is this preventative strategy. Can we actually expose a plant to a material that will induce stress response and prepare the plant for a stress that's actually going to happen in the future? And when you think about climate resilient agriculture, that's going to be something that's going to be interesting. But taking a step back, as I said, we got a lot of this retrospectively looking at our data. So in 2014 is when we started working on using nanoscale micronutrients as a means to induce tolerance to biotic stress, to pathogens. So we started working with fungal pathogens. In the US, we spend about $600 million a year on fungicides, and we're still losing about $200 million in revenue to fungal diseases. So we started considering micronutrients because these are the elements, these are the nutrients that are really important to these stress-related pathways. But we know that there's difficulty in getting sufficient amounts of these into the tissues where you need them. So for a soil-borne pathogen that's going to infect by the roots, you need these micronutrients in root tissue. So that's where we have to get them. So we actually started asking, well, what if we supply these nutrients in nanoscale form? We know nanomaterials are smaller, they're more active, they're more reactive, they're more mobile. So we aren't, even though we're working with disease systems, we're not targeting the pathogen directly. We're targeting this plant response, this stress-related response. So this was the first paper we published in this space. We published it in 2016 in a journal called Environmental Science Nano. We work at an agricultural experiment station, so we want to come up with strategies that farmers are actually going to use. So our strategy was pretty straightforward. We do a foliar application to seedlings, and then the seedlings are transplanted to the field. That's how a lot of agriculture actually happens in New England in the United States. So we looked at different micronutrients, copper, manganese, and zinc, in different forms, the nanoscale form and the conventional forms. So we're adding 100 ppm, which sounds like a lot, but it's a foliar application. So we're only adding a couple of milliliters, which means we're well below a milligram of these nutrients. So, and the short of the long is what you see is, so this is field data. This is eggplant and tomato under infection with Fusarium, which is a fungal pathogen. Focus on the green because that's the copper. And what we saw was, in the middle green bar is the nanoscale copper. With that foliar treatment, months later, we're getting significantly greater numbers of fruit being produced per plant. This is eggplant data. This is a measure of disease, so we're getting significantly less disease, and we're getting much more copper, well, significantly more copper in the roots. And I'm not showing you the data, but the alleviation of stress and ROS specifically what was implicated. So because this is a field study, we knew that we spent about $44 on nanoscale copper. That's what our cost was for the material. And in terms of yield, we were increasing economic return for the farmer by about $10,000 per acre, or the way I prefer to think of it is in terms of increased food that we're producing. So one of the other, as I mentioned, I'm in a National Science Foundation funded center. It's called the Center for Sustainable Nanotechnology, and there's a significant initiative in that center. The center runs for 13 years, and we're just starting our 12th year now, so we're almost done. But there's a significant effort in looking at how we can control the chemistry of these micronutrients to optimize the benefit, optimize the recovery from biotic stress in this case. So we took commercial copper oxide nanoparticles, which are just the same materials we were just looking at. And then we also made a material where we changed the morphology, we made it a sheet, we changed the composition, we added a phosphate. We wanted to see how tuning that chemistry would alter the response. And so this is another field study. This is with watermelon and another Fusarium pathogen. This paper was published in 2018. Same exact strategy, foliar application to seedlings, then the seedlings go out into the field. And in terms of disease response, what you see is the yellow is the commercial material. You got to get up to a couple of 100 ppm to see a decrease in disease, or yellow down here, an increase in biomass. But look at the dark green bars. That's our custom copper phosphate nanosheet. At a 10-fold lower concentration, we're getting a significant reduction in disease and a significant increase in biomass. So by tuning the chemistry of your material, you can dramatically increase its efficacy. So this was the paper that we really kind of worked out this mechanism relating it to reactive oxygen species and stress. We published this in Nature Nanotechnology at the end of 2020. We switched to soybean and the Fusarium that affects soybean, which causes soybean sudden death syndrome. This is just a greenhouse study, but we looked at a couple different kinds of custom copper nanomaterials, because again, this is work done within the center. And so this is just a greenhouse study, but what we see is that when that fungal pathogen is present, you get about a 60 to 70% reduction in yield, photosynthetic output. You see significant oxidative stress as measured by a number of different parameters. But importantly, the nanoscale copper treatments significantly alleviate that stress. They increase biomass. They restore the levels of photosynthesis and other damage. So what we did here was this was the first time we really started looking at gene expression. We looked at almost 24 defense and RLS related genes in the roots. So we're doing a foliar application and we're seeing the response in the roots, and all of these pathways were significantly upregulated. But what was interesting is the extent to which they were upregulated was a direct function of the properties of the different types of copper that we were using. And we were actually able to describe that computationally. There's a computational chemist group in this center. And that's what ended up getting us the cover, I think, of this journal. So it's not just all about copper. One of the other materials we're looking a lot at is sulfur. And this is a lot of work we're doing in collaboration with Professor Owen Pargash-Danker, who's sitting in the first row here at the University of Massachusetts. So this is a USDA-funded project. This was a paper we published in ACS Nano at the end of 2022. And in this case, sulfur is really interesting because in the United States, 25 years ago, nobody cared about sulfur because there was enough sulfur coming down in acid precipitation that there were no deficiencies in soil. But we had the Clean Air Act, which was passed, and now many farmers in the United States have to add sulfur to soil as an amendment. The problem is when you add sulfur to soil, you get significant changes in pH in the microbiome. So we were interested in what would happen if we added that sulfur in nanoscale form. And again, this is specifically in disease systems. So we looked at a coated and an uncoated form. This is tomato and a fungal pathogen. My postdoc on this, who's now a staff scientist with us, measured all sorts of endpoints over about a 2-week exposure period. So it was a time-dependent harvesting of these plants over those 2 weeks, looking at basically every parameter you could think of, including some time-dependent gene expression and metabolomics by high-resolution LC-MS. So in terms of what we saw, when tomato is infected with Fusarium, we get an 87% decrease in biomass. This is just a greenhouse study. I'll show you the field study in a minute. But what we saw was our 2 nanoscale forms of copper or 2 nanoscale forms of sulfur were the most effective at reversing the impacts of disease, increasing biomass. The other forms of sulfur actually do pretty well, but as I said, those other forms of sulfur are what are causing all the problems in soil. So what we noticed was that when you looked at the ratio, looked at the distribution of sulfur in the different tissues as a function of treatment type, and in this case, this was a soil treatment because, as I said, that's how growers are adding it. But we saw particle size specific translocation ratios. We looked at 2-photon microscopy and gene expression, and it really looks like when you have sulfur in nanoscale form, you're actually using a different uptake pathway. You're not really using the sulfate pathway, which most plants are using. You're using this elemental pathway, which ends up being much more efficient for the plant, particularly under disease. So the time-dependent gene expression and time-dependent metabolomics ended up being really interesting. As I said, I don't know if you can see this, but we're harvesting plants at 4, 8, and 16 days. And what we saw was that, and this all was a function of nanoscale treatment, that the gene expression really wasn't impacted at 4 days. Many of the stress-related genes came up, were upregulated at 8 days, and then dropped off at 16 days, whereas the metabolites, there was a delay, obviously, because of the central dogma. We didn't really see much metabolites response until we got to day 16. But again, the pathways that were most impacted, that were most interesting, were all of those surrounding stress and reactive oxygen species. So this was a field study. It had the exact same design as that ACS Nano paper we were just looking at, except none of the endpoints. The only thing we were looking at was yield because it was a field study. And this was published in 2022 as well. But the results were basically the same. We got a significant increase in disease suppression and increase in biomass when we did this nanoscale sulfur amendment. One of the things that we also see, and we've seen this across systems now, is we see essentially a nano-enabled biofortification of the edible tissue. When you upregulate these pathways, many micronutrients tend to increase, and we suspect many organic nutrients are as well. But again, here's the math, here's the practical aspect of this. This is how you sell it to a farmer. We're only spending about $33 an acre on nanoscale sulfur. And in so doing, you know, we more than doubled the yield of tomato per acre in a healthy system, from 4,900 kilograms to almost 12,000. And even in the disease system, we're still doubling our yield, and we're doing this in the complete absence of any fungicides at all. So it's not all just about biotic stress, we're also interested in abiotic stress and how you can control ROS homeostasis to alleviate that. And again, we were just hearing a lot about abiotic stress. This is a paper we published in 2019 where we're looking at drought. So in this case, the D is not disease, it's drought. And we're looking at zinc, nanoscale zinc. This is sorghum. So when you expose zinc, when you expose sorghum to drought conditions, you see a decrease in yield and many of these other important parameters. But if you have supplied nanoscale zinc in the soil at relatively low concentrations, many of these negative impacts are reversed. And this is all a function of the zinc being accumulated in the plant and upregulating these stress-related pathways and helping to alleviate the ROS damage that the drought condition is causing. We see a similar situation with cerium. And in this case, the abiotic stress we're looking at is salinity. Now, cerium is interesting because it's actually not a nutrient. It's not required, but it's a known antioxidant. So again, honing right in on ROS homeostasis. In this case, we used cotton because we had money from the cotton board, so we had to use cotton. And it was a seed treatment in this case with a coated cerium oxide, but the same exact thing. When you expose those germinating seeds to salinity stress, if they were pre-treated with that nanoscale antioxidant, and I should mention all of these effects that we're talking about are nanoscale specific. We always have the conventional controls, the bulk and the ionic, and they never work, or almost never work. So when that nanoscale cerium is present, you get increased growth, you get increased root vigor under the stress. When you actually measure the ROS, you find less ROS if the seeds were pre-treated with cerium. And when you look at what was being increased in terms of gene expression, it's all of these same pathways that we're talking about. Excuse me. So we have a lot of work going on in this space. This is just papers we published in the last 2 years, looking at different micronutrients for the most part, and how they can impact ROS homeostasis under a variety of conditions, stressed, non-stressed. You just see some of what we're looking at here. Molybdenum, because it's for molybdenum and nickel because of their impact on nitrogen fixation, magnesium, lanthanum. What we've also started to do is look at more green or biosynthesized nanoparticles. This is a topic that's not very popular or not looked at very much in the United States, but in lots of other countries, including India, biosynthesis of these nanomaterials is quite intensely being studied. And the reason is you get a different kind of material. You don't just get nanoscale selenium, you get nanoscale selenium with all of these biomolecules associated with it, and it behaves differently. So, as I said, lots of interest. So I'm just going to finish on the reverse. So the first half, or the first 2/3 of this, was you have a plant, it's exposed to a stress, disease, salinity, drought, and you can supply nutrients in a nanoscale form, and you will get a nanoscale specific alleviation of that stress through ROS quenching. So can you do the other thing? Can you actually pre-treat plants or seeds with a material, a nanoscale material that's not going to be overly stressful, that's not going to cause damage, but it's going to be, the dose is going to be appropriate to actually just upregulate all of these pathways. So then when that plant later, weeks, months later, is exposed to a stress, it's almost like a vaccination. It is prepared to handle that stress. So this was one of 2 papers we published in this space. This was published in ACS Nano last year, at the end of last year. And The nanomaterial that we're using to induce stress is silver. We know this causes an ROS response, so what we need to do is we need to find a dose that's low enough that it doesn't actually cause toxicity, but it's high enough that it upregulates all these pathways. And in this case, we're using rice. So we call this stress training. We train them as seeds or seed priming or leaf priming, or together seed and leaf priming. And so then those trained seeds are grown, and then weeks later, they're exposed to either rice blast or cold stress. And then we do the metabolomics and the transcriptomics to figure out what's going on inside of the plant. So I know I'm almost out of time. I'll finish quickly. So this is the data. So up here is the disease response. So this is just lesion size on the leaves. And what you see is regardless of how you did that stress training, when the rice is exposed to that fungus, literally weeks later, you see significant reductions in disease across the board. And the greatest reductions in disease are when you do the seed priming and the leaf priming together. You're looking at a 70, 80% reduction in disease presence. Here's your cold stress data. It wasn't as dramatic, but still you see an increased tolerance with the seed and leaf priming, increased tolerance to cold stress. Again, and this is cold stress that's literally happening almost, I think it's 28 days after the treatment. So I won't dwell on this too much, but as you may suspect, all of the pathways that are being upregulated are these same exact pathways. They're all related to ROS scavenging or stress response. And you see that in terms of gene expression, and you see it in terms of metabolites. So the second study, which was just published in December, we switched to corn. We switched to our stress-inducing agent. We included silver again, but we wanted to switch to an actual nutrient so we could maybe get some additional benefit. And then the other thing was we looked at single stressors, but we also looked at multiple stressors together. So if you prime a seed with a corn seed with iron and then expose that corn plant a month later to drought and cold or drought and saline conditions. When you looked at the single drought conditions, corn behaved the same way as rice did. You get that stress alleviation. You get that priming effect. And you get it with not just silver, but you get it with iron. But what's really interesting is when you look at multiple stresses, you see increased tolerance, again, weeks later to multiple stresses. And what's really interesting, we haven't published this yet, is this seems to be transgenerational. We actually collected the maize seeds, even though we published this paper. We collected the maize seeds and grew them out under stress conditions in the F1 generation, and they're still exhibiting some of this tolerance. So hopefully in 1 minute too long, I convinced you that nanotechnology does have significant potential here. There's plenty of ways to use nanotechnology poorly in agriculture. I think we're working on some ways that are actually safe and sustainable and can exert really significant benefits to plants. And being able to control that ROS homeostasis, control when and how those pathways are upregulated, really you can have some dramatic effects on yield under stressful conditions. So we've got a very large group of collaborators, and I listed most of them up there, and Om sitting in the front row is one of my best collaborators and best friends, so it's always good to come to a meeting with him. But I will stop there.
Thank you, thank you. Very, very interesting lecture.
Right before lunch.
Bharat Prasad from Kansas State University. Very nice presentation, Jason. High quality publications and supported with the data. So my question is related to the stress memory, which you talked about at the end of your presentations. So you slightly touched about the transgenerational things. I was wanting to know if there is a memory. Does these nanoparticles uptake go into the seed, and then it still keeps the memory, and in the next generation they're going to provide stress tolerance, whether it is biotic or abiotic stresses, kind of epigenetics of this stress and nano interaction?
Yeah, as I alluded to, and we've only done the experiment once, so it hasn't been replicated yet, but those maize seeds, when they were exposed to subsequent stresses F1 generation, there was an equivalent level of tolerance to single stressors. We didn't look at multiple stressors, so there's clearly something going on. But you raise a good point about the nanomaterial itself, because a nanomaterial of copper or sulfur or iron is completely useless to a plant. The biologically active form is the ion, right? I mean, that's what's needed. So there's a significant mechanistic gap in our understanding. We are supplying these nanoscale nutrients either as a foliar treatment or a seed treatment, or in all these different combinations. And we know under certain conditions there are some nanoparticles getting into plants, but a nanoparticle in a plant is not going to give a plant any benefit. So that's when this chemistry of the material becomes really important, because you can tune its dissolution. I've got a collaborator at the University of California. He actually just published a paper in Small where he took carbon dots, and when he coated them with sucrose, he could actually put the carbon dots on the leaf, and he got 70% transport of the sucrose-related carbon dots to the roots, because the phloem transporters were recognizing the sugars. So if you really need to load something into the roots, coat it with a sugar, because that's what's going to be translocated. So this ability to understand the chemistry, I think, is really going to be critical, because you can control where the material is going to go.
Quick follow-up. So the way it is manufactured, you said, has an impact on the biological activity. So if you were using the biological ways of manufacturing these nanomaterial, will it have a more transgenerational positive impacts? That's number one question. Second one is, have you looked at the translocation of these nanoparticles, fluorescence are they ending up in the seed to have an impact on the next generation or not?
Yeah, so to answer your first question, the transgenerational impacts with the biosynthesized nanoparticles, I think so, because the little bit of work that we've done so far does suggest that when you take that nanoparticle and generate it by a biosynthetic process, in some ways you get a completely different behavior because of all of these polyphenols and all these plant metabolites that are associated with it. So I would suspect if you're going to anything that you're going to see with a chemically synthesized material, I think there's a pretty good chance it's going to be greater with a biosynthesized one. We have no data to support that yet, but that's just my hypothesis. The second question, which I just forgot. The tracking. Yeah, we have tried to do that. In the 2 sulfur studies, we used 2-photon microscopy. And sure enough, when you put sulfur nanoparticles in the soil, you can see sulfur nanoparticles coming into the roots. And 2-photon microscopy is nice because it's a non-destructive process. So we actually have videos. We have movies where we can watch the particles coming in. That's another reason we're using carbon dots, because confocal fluorescence microscopy can be used to track those. So you definitely do see particles moving into plants, which is generally something you want to avoid or minimize. But in some cases, that might be what you need, because if you're doing a soil treatment and you want these materials in the leaves, then the best way to get that is have the particle move all the way and then dissolve when it gets there. But you need to be very careful, because if you start loading nanoparticles into rice grains, there's a lot of people who are going to be upset that you've loaded nanoparticles into rice grains. So that's why, way back in the beginning, that's why we started focusing in on micronutrients, because we knew if we saw a benefit, the benefit was not because of the nanoparticle itself. It's because the nanoparticle enabled us to get to a better location or to do something different, but it's still all about the dissolution. I've got collaborators who are using carbon nanotubes in agriculture and still want to use silver. I think that's a terrible idea, because they're not materials that are natively found in plants. So it's inherently a higher risk. And if the risk is higher, the regulatory community is going to look at it with greater scrutiny as well. They should.
So the next session is by Dr. Ajit Kumar Shashani, Director, CSIR National and Botanical Research Institute, Lucknow, India. We'll be talking on the topic Metabolic Engineering for Climate Resistance Agriculture. This would be on the video conference. Please watch on screen.